Good evening. We'll try it again. Good evening. Thank you, and welcome to the Shoals Marine Lab's weekly science seminar series. I'm Sarah Morris, the John M. Kingsbury Executive Director of the Shoals Marine Laboratory, and I would like to welcome everybody today who is joining us for our 9th Rock Talk of the 2026 season. For anyone who is new to our lab, the Shoals Marine Lab is the largest and oldest marine lab in the country that is focused on undergraduate training, both in research and in coursework. The lab is operated jointly by the University of New Hampshire and Cornell University. Our talks are affectionately called Rock Talks because back in the day, they literally happened out on the rocks. We did not have walls or windows on a number of our buildings at that time. Now that we've migrated indoors, we still keep that name as one that's a throwback to the early years of the lab. And this provides an opportunity for everyone in the lab to come together to hear topics of general interest related to marine biology, conservation, environmental science, and other related topics. In addition to our guests who are joining us remotely tonight, we have 3 classes on the island. The first is Shark Biology and Conservation, Marine Environmental Science, and Research in Biology. In addition to our 3 classes, we also have our 10-week undergraduate research interns, affectionately known as Surges. We're excited to have everyone joining us this evening, both in person and online. The format for tonight's talk is that we will have a 45-minute presentation followed by about 15 minutes of questions and answers. For those of you joining us online, please use the Q&A button at the bottom of your screen to ask questions. For those of you in the room, I will be coming around with the microphone so that the people online will be able to hear the question in addition to the answer. This evening's speaker is Dr. Sarah Joy Bittick. Sarah Joy completed her BS at UCLA in marine biology, and she continued at UCLA for graduate school, where she received her PhD in 2017. Her dissertation examined the interactive impacts of nutrient enrichment and overfishing in coral reef and seagrass ecosystems. From 2017 to 2019, she held a Liber Aero postdoctoral fellowship at the University of British Columbia, and her research at UBC focused on informing local seagrass management through studies of nutrient input and invertebrate community structure. She's currently an associate professor in the biology department at Loyola Marymount University in Los Angeles, where she leads the Coastal Ecology and Conservation Lab. Her research focuses on human impacts in nearshore marine ecosystems, including seagrass meadows and rocky intertidal habitats. And she works alongside a large and enthusiastic group of undergraduate researchers. At LMU, Sarah Joy also teaches general biology laboratory courses, marine biology, coastal ecology, and marine conservation. This evening, Sarah Joy will be presenting the impacts of herbivory and nutrient supply on macroalgal defense traits. And I would ask you now to join me in welcoming Sarah Joy Bittick.
Thank you for the introduction. And I heard I had just missed the phycology professor who was teaching here in the last block, but you're going to hear a lot about macroalgae today. And as Sarah mentioned, I'm coming to you from Los Angeles, so almost as far as you can get away from Maine, and still be in the US. Loyola Marymount University is a small private institution, undergraduate focused. So all of the research that I've done at LMU is with undergraduates, such as many of you. So a little bit of an overview on where this talk is going. I'm not going to assume everyone knows what macroalgae and seaweeds are. There are lots of shark people here, bird people. So we'll do a brief intro to what macroalgae are and how nutrients and herbivory interact to affect the abundance of algal species. And what I've been studying at LMU, and actually since my PhD, are the defense mechanisms that macroalgae have to defend themselves against herbivores. In the tropics, this is usually herbivorous fish. In temperate environments like here and off the coast of California, more invertebrates, crabs, amphipods, isopods, things like that. So my body of work in this realm started at UCLA, actually, as an undergraduate on a marine biology quarter field course, similar to what a lot of you are doing. And then I continued to study this tropical species of macroalgal or seaweed in French Polynesia. And so this kind of set the groundwork for the interactions that I look at with the temperate species that I want to tell you about, because it has some similarities to species that you'll see here off the coast of Maine. So I'm going to share briefly a couple of studies from French Polynesia first, coral reefs, and then I'll get into the temperate macroalgae research, Silvetia compressa, the rockweed that we have in abundance in Southern California. And if we have time, professors like to talk a lot, but if there's time, I'll tell you about some of the ongoing research my students are doing and our next steps. So if you don't know, algae are not plants. They photosynthesize and they hold similar ecological roles, but they don't have vascular tissue. This kind of classification, kingdom Protista, just is a conglomerate grouping of things that aren't plant animals or fungi. I also study seagrasses. Unfortunately, I don't get to talk about that today, talking about my algae work, and specifically macroalgae, as opposed to microalgae like your phytoplankton. And so they are superficially similar morphologies to plants, basic anatomy that you'll hear me talking about a lot. The whole individual is called a thallus. And they have blades that look like leaves and stipes that look like stems, but they aren't true leaves and stems. There's no xylem and phloem for nutrient exchange or anything like that. Every cell can kind of absorb nutrients. And their hold fast system is simply to hold them onto the rocks, not to exchange nutrients and gases. But they hold these similar ecological roles to plants, in some cases forming even macroscopic forests, like in the case of kelps off the temperate coast. So we're not talking about the microalgae, we're talking about the seaweeds, which can fall into the groups of brown, red, or green macroalgae. And specifically, we're going for the browns, the rockweeds. And these species tend to be slower growing than our green algae. So I saw some out there, went down to the intertidal. There was some ulva species, these more fast growing ephemeral species of algae that respond really quickly to nutrients. And this is what I call a bottom up process. So you input nutrients and the algae grows. But with our browns, like Turbinaria in French Polynesia, or the Silvetia that I study off the coast of California, they don't grow very quickly in response to nutrients. Obviously, they need some amount of nutrient input to grow, but they're persistent species that grow very slowly over time. So as opposed to growing fast in response to input from nutrients, maybe they're using it for something else. And that's what the majority of my talk will be about. On the other hand, some of you are studying top predators. So when I'm thinking about top-down interactions, I'm kind of going as far as the herbivores, and that's top-down because they're having a top-down force on the macroalgae. But we're thinking about herbivorous fish in the tropics, and as I mentioned, things like snails and crustaceans in temperate environments. And so typically those fast growing species like the green ulva, those are more palatable than our slow growing brown algal species. And part of the reason for that is these anti-herbivory defenses that are abundant in brown. So typically these two pressures, bottom up and top down, interact to influence the overall abundance of your primary producers. Something's coming in and eating the primary producers. Nutrients are the resource. And you would kind of expect that when you have both of these things happening at once, say nutrients increasing because of human pollution, and the pressure of herbivory decreasing, that algae might explode in abundance, right? Because nothing's there to eat it and the nutrients are causing it to grow. But a lot of interaction work looking at nutrients and herbivory, especially in tropical ecosystems, have found that as nutrients increase, it increases the nutritional value of the alga. And so fish eat more of it. They eat more of the nutrient-enriched algae. However, our brown species, remember these are those slow-growing, persisting species, they also tend to be pretty unpalatable. They have a lot of different defenses, both physical and chemical. So by physical defenses, I just mean mostly toughness. So they end up being really strenuous for herbivores to consume them. Chemical defenses, there's many different chemicals that can lead to herbivores avoiding macroalgae, such as polyphenols. And there's a couple that found polyphenols that are found in brown algae. They're characterized by the carbon rings and the OH groups. So silicon, silicon basically embedded in the tissues of the alga. This is really well researched in plants, in terrestrial plants, where the mandibles, the insects that are eating the plants in this case, are worn down by silicon embedded in the tissues. Algae have this as well. It would be akin to this, I have my toddler around here. There's this, that's not my toddler, but this little guy with a mouthful of sand. It would be really similar, glass, sand. So these are some of the defenses that brown algae have been shown to have to defend them against herbivores that may, come and munch on them. And the body of work related to this is pretty well established for a few species, like Turbinaria ornata, the species that I've studied in French Polynesia. But even before that, groups of French scientists found that nutrients increased the toughness and phenolic concentration. So toughness goes up, phenolic concentration goes up. Nothing really wants to eat this Turbinaria ornata. And that herbivores further that toughness. Herbivore comes in, munches on it, and the alga increases the toughness even further. But on temperate coastlines, few species, which you also have here and we have in the Pacific, also has been shown that herbivores can induce those defenses. But we don't, and I also found a paper from Sweden on Ascophyllum that you have abundantly out here, and I touched it for the first time today. So I study Silvetia compressa on the Pacific coast of North America, and we don't know much about its chemical and physical defenses. But these algae are all in the same order. They're all bucales. And as I mentioned, I had to go down to the Great Tide Pool and so touch Ascophyllum. It's quite similar to the Silvetia compressa that I'm studying in Southern California. And I'll show you more pictures of Silvetia compressa on the Rocky Intertidal. But first I want to set the stage by showing you some work from the South Pacific with that species Turbinaria ornata, because this is where kind of the inspiration for all of the nutrient interaction and herbivory started. And just to give you a little bit of background about tropical macroalgae specifically, they tend to be very small or heavily defended against herbivores because you have so many fish in herbivorous fish in the system. And so there aren't a lot of nutrients in tropical ecosystems. So when you do get these pollution events with lots of nutrients, it can be a problem. But typically there's high herbivory of fish by fish and urchins, and the algae are kept pretty small. So that of course is under undisturbed conditions, right? And I did this work on Turbinaria ornata in French Polynesia. So now I'm taking you all the way across, almost across the Pacific, basically surrounded by ocean, as far away from a mainland as you can get. And there had been a series of disturbance events in Moorea, French Polynesia, one of which was this crown of thorns sea star that actually eats the corals. But this disturbance, among other things, led to this algae Turbinaria ornata growing in abundance. And it is a brown algal species in the same order as these temperate species that I study, and it's heavily defended against herbivores. So it's tough, it has those polyphenols I mentioned, and it's been expanding across the South Pacific and the Caribbean for reasons that are native to those areas, but becoming a nuisance species. So my first research question was to look at how human manipulations of nutrients and herbivory, so nutrient pollution and overfishing, may influence how Turbinaria has been expanding across the South Pacific. And I'll only be able to share really one study question with you that looked at whether nutrients and herbivores, whether there's an interaction between those two. Remember, I told you that we typically expect with more nutrients, herbivores will eat more of the algae because it's more nutritious. So we did a pretty simple experiment at a field lab like this in Moorea, French Polynesia, where nutrients were added, a fully crossed two-factor experiment with nutrients added or left ambient. So minus nutrients means no nutrients added, plus nutrients means we added a little Osmocote baggie, which is just nitrogen, phosphorus, slow release. And then we caged half of those and left half of them out. So your most normal treatment would be herbivores have access, so there's no cage and there's no nutrients. And we also kept out seagrass and conducted this on a shallow French Polynesia. So what we found was not the hypothesis that I just told you about. What we actually found was the opposite, that when you have nutrients, fish can't eat the algae. So here we have plus nutrients plus herbivore means we added nutrient, and the herbivores could the algae, they had access to the algae. The algae is growing. It's not, there's no net consumption. But when you take away the nutrients or leave it ambient and the herbivores have access, now they are able to. So the herbivore only eating the Turbinaria that is lower in nutrient concentration. So at this point, I thought my dissertation was ruined because it wasn't what I hypothesized. But it actually ended up being even more interesting. So some words of wisdom, develop your research. And what we noticed as we were measuring the biomass of the algae was that the ones that were nutrient enriched were really, really tough and spiny. We're out in the middle of the Pacific Ocean with not a lot of fancy lab equipment. So we measured toughness using a pen and a cup and put incremental amounts of weight as it lay over one piece of the alga and watch it until it just pierces it. And we found that when nutrients were added to the algae, it was much, much tougher. It took a lot more weight to pierce that piece of algae. And when I ground up algae, put it in agar plates, agarose, fish will eat it. They ate the nutrient enriched algae. So in the absence of that physical defense, they preferred the nutrient-rich type. So pointing you to here, this is in these agar plates. Now a lot more is being eaten by the fish. So in the absence of physical toughness, the herbivores did eat nutrient-rich algae. So it's not that the algae isn't becoming more nutritious, it's that it's also becoming tougher. And for the sake of time, I'm not going to show you a newer result that kind of confirms that herbivores are able to compensate for increased toughness of Turbinaria ornata in deeper areas where the fish are larger, which could be good news for the resiliency. So if you want to hear more about that, I can tell you that. But I want to make sure that we get to the Silvetia work. So what we found with Turbinaria ornata was that there is this interaction between nutrients and herbivory, but it was not in the direction we initially expected. It was in the direction that nutrients were increasing toughness, thereby causing the herbivores not to eat the algae because it was so tough. And because of lab strains out on the island and having no export permits from French Polynesia, we weren't able to measure the chemistry, but there is some work that's been done showing that phenols do increase with increased nutrients in Turbinaria ornata. So this brings us to my temperate ecosystem, the rockweed Silvetia compressa that we have on the Pacific coast. So just to mention a few things about temperate macroalgae, they're generally larger due to cooler water and more nutrient availability, but our herbivores are much smaller, as I mentioned. Mostly invertebrates, some small fish. Some are defended, and it's been shown that they're defended against herbivores, but there may also be an interplay of using these chemicals to help tolerate certain abiotic conditions, which I'll get to. So there's some of our greens, some of our reds, and then we have our kelps that make up the kelp forest. But what I'm seeing is the rockweeds. And in California, we have both fucus, it's the stickus in our neck of the woods, and Silvetia. And to show you again, but you can see it looks pretty similar to our Silvetia. Maybe you can't see as good in that picture. I think I have a better one later. But in this case, in the temperate environments, there tend to be a lot of nutrients, or a lot more nutrients at least, than in the tropical ecosystems, but we still can get these blooms. We get these blooms of typically green algae, and these fast-growing ephemeral species tend to take over the more complex browns and seagrasses and salt marsh plants. So in French Polynesia, we had this situation where a brown persistent species was taking over the natural foundation species. And on the Pacific coast and here on the East Coast, it's typically green algae, some reds, that outcompete the browns. So browns are good over here, right? There are foundation species. In Southern California, we have a lot of Silvetia compressa, and small herbivores do eat it. So there I am showing you a grazing scar. And similar to that Ascophyllum, Silvetia grows all over the rocks. And it's found in the mid-intertidal to high intertidal from Baja California all the way up California's coast, almost to Oregon. It's highly productive. Not only do some organisms eat it, as I've mentioned, but it forms this foundational habitat for other seaweeds and lots of invertebrates. And in Southern California, it's been on decline due to human trampling and in favor of smaller, less productive algae. But this entire order of brown algae Bucales is known to have chemical defenses. So my goal with my research team at Loyola Marymount University was to employ some of these techniques to study whether top down, bottom up, so herbivory and nutrients, whether these factors interact, work independently to influence our Silvetia compressa population, either its biomass or its anti-herbivory defenses like toughness and chemistry. So as I mentioned, Loyola Marymount University is a predominantly undergraduate serving institution. And so this work couldn't have been done without a number of undergraduates from the university. This first iteration of the project started kind of around this time of the pandemic. And I had some students who were very persistent in working through those times and continuing to work on this research with me throughout the years, conducting their senior capstone research in the Rocky. So I'm going to kind of break the bottom up and top down factors independently first. So one question we had was whether nutrient enrichment impacts the growth rate of Silvetia compressa tissue. Remember with Turbinaria ornata, we didn't really see nutrients causing it to grow a lot more. We saw nutrients being used to make it tougher and possibly to increase those phenolic defenses. And so with that, the second question, do nutrients affect the chemistry and physical defenses of the algae as they did with Turbinaria? So I want to just orient you to Los Angeles. So this is the greater Los Angeles area. Loyola Marymount University is right by the airport, pretty much. And we do a lot of our work down in the Palos Verdes area, which is beautiful. The rocky intertidal here looks similar to the Great Tide in a way. But instead of aspergillum, we have Silvetia compressa. So you can see the similarities. It doesn't have the same nematocysts, but it serves a similar ecological role, providing a foundation for lots of organisms on the rock, in the rocky intertidal. And so my students used similar techniques to the Turbinaria study, just measuring initial biomass and the physical toughness. And then in a tank experiment at LMU, we have a vivarium with full spectrum lighting and temperature controlled chillers and everything. We manipulated the amount of nutrients in the tanks. So basically just different concentrations of nitrates and phosphates, from a lower ambient concentration to a higher level at 5 micromolars of nitrate and 0.5 micromolars of phosphate. And after growing it in the nutrients, we then measured growth and toughness again. We saved half of our samples for analysis of all of the chemistry. So this is the phenol analysis using a Prussian glue reduction method and X-ray refractometry for the silicon. So I'm not going to show all of those results because I want to get to the hermit crab herbivory experiment. But in this case, nutrients did increase toughness like they did for the Turbinaria, and nutrients had an effect on phenols. It wasn't completely directional. Our 4 micromolar treatment was the one with the highest concentration of phenols, and silicon also increased in response to nutrients. So we didn't really see an increase in growth rates, just like Turbinaria. Again, persistent species not growing super fast. All of these characteristics increased in response to nutrient enrichment. So that was after 10 days of being in the tanks. Then we threw in threw in hermit crabs in different treatments. But just to summarize what was happening with the nutrients independently, there wasn't a significant impact based on biomass. So not really using the nutrients to grow really fast, but nutrients were causing an increase in toughness and having an effect on the phenol and the silicon. So what about the herbivores? What about that top-down part? Do herbivores prefer Silvetia compressa of a certain nutrient status? So this is kind of that same question, like, are they going to eat the nutrient-enriched algae or not? And do hermit crabs impact the anti-herbivory defenses? And now, because we have this second factor of herbivory, we can run tests to see if nutrients and herbivory, those factors interact with each other, leading to a difference in algal responses. So after the 10 days of nutrient enrichment, we added in herbivores in 3 different treatments. There was a no herbivory treatment with 0 hermit crabs, and treatments with 15 hermit crabs and 34 hermit crabs. It was just the amount that my students were able to collect that given day. No significance to the 15 and 34, just to have a gradient. And then we did the same measurements of traits. We measured whether there was growth or consumption by measuring biomass, and we measured the toughness of the algae, and then later we measured the biochemistry, the phenols and the silicon. So first, to show you what's going on with biomass and toughness, herbivores. So here on the y-axis is going to be my biomass change over the 10-day period. Nothing was quite significant here. We ended up with only 5 algae in each algal balli in each treatment by the end, because we sacrificed half of them to measure the phenols between the experiments. But you can see that the herbivores are eating the Silvetia, and there's a trend towards them eating 4 micromolar. So there is, again, not quite direction. It's not quite that they're eating the highest nutrient enriched. There's a sweet spot with the 4. And so, low sample size, marginally significant. There seems to be some sort of possible interaction between nutrients and herbivory that's impacting the net biomass of the Silvetia. So this is what we have as far as our Silvetia compressa information compared to our Turbinaria ornata information. There's probably an interaction here between nutrients and herbivory, and we know that all of our defense traits are increased by nutrients. Okay, so what about herbivores and the interaction between herbivores and nutrients? Is there any impact of adding herbivores to this system that may cause a difference in these algal defenses? So that's where we get back to our toughness test and our biochemical assay to measure phenols and so first, looking at that puncture method again, we saw that herbivores overall are decreasing the toughness. There is this 4 micromolar that's kind of behaving differently in the 0. So this is no herbivores, and that 4 micromolar is still able to basically increase its toughness with the nutrients that it was supplied. So the herbivores are eating the algae and weakening its cellular structure. So since this is just only the one factor of herbivory was significant, I'll just show you this to simplify it. Herbivores decrease tissue strength. They decrease toughness. If you remember back to Turbinaria, That was the opposite. Herbivores actually increased. So herbivores decreased toughness, and we didn't have strong evidence for an interaction. That's why I'm showing you this one. So different than Turbinaria in some ways. Nutrients increase the toughness and herbivores decrease the toughness of the alga. And then looking at the biochemical assays with the Prussian blue, basically it's a reaction using iron and breaks apart those OH groups that are on the polyphenol, turns blue, and you can measure it with a effect. And in this case, only nutrients were significant. So again, instead of walking you through the two-factor plot, I'll just show you the one-factor plot. And again, we're seeing nutrients impact phenols, but it's not a straight, like, directional. So there is some impact of nutrients on phenols. But we saw in this case that so we're measuring absorbance, basically those blue colors. And the absorbance in this case was highest at 5 and 3 micromoles. And lowest at 2. So herbivores don't really seem to have an impact on the phenols. In this case, it was, as I'm showing you here, only nutrients. So no impact of herbivores on phenols, which for a reminder that for fucus, herbivores have been known to induce those chemical defenses. So it may not be true for all rockweeds. There's some species specificity. And there was only that nutrient. The nutrient. Okay, so I've talked a lot about algae getting tougher or having more or less phenols, but I mentioned to you silicon embedded in the tissues of, it's well researched in terrestrial plants, less up in macroalgae, but silicon is present in macroalgae. And so we decided to measure the concentration of silicon in the tissues of Silvetia compressa. And in this case, found that both nutrients and herbivory were affecting the silicon concentration, but they weren't interacting. So again, I'm going to go from this two-factor plot with our herbivores on the X and silicon concentration on the Y-axis to the one-factor plots that are more clear. So silicon on our Y-axis with nutrient enrichment, a clear increase. So as nutrient concentration increased, silicon concentration also increased. And it's the opposite with the herbivores. So as herbivore density or herbivore load increases, silicon decreases. And my thinking here is that the herbivores are able to bite through and basically break apart the silicon tissues, or basically degrade the tissue of the macroalgal. So this hadn't been shown before, that nutrients can potentially, nutrient enrichment can potentially increase silicon concentration in macroalgae. But we're seeing that that nutrients are increasing silicon here. So we see that our herbivores decrease the silicon concentration, but that those things aren't interacting with each other. So to summarize the Silvetia compressa responses one more time. Similar to Turbinaria ornata, we're seeing an interaction between nutrients and herbivory, but it's kind of in a different direction. Nutrients still increase toughness, but herbivores decrease toughness. And I'm starting to think this is all related to the silicon, but this is like new in my thinking because before I was really focused on the phenols, like it's about it's about, yeah, I mean, silicon is an element also, but it's physical. It's like a physical defense because it's embedded in the tissues. So I think that silicon might be very important actually in these algal systems in deterring herbivory. So I have a few more minutes. I can't show you all of the ongoing work because this kind of opened up a lot of questions because Silvetia compressa isn't necessarily responding similarly to the East Coast and Baltic Sea fucus. And so we're still kind of working out these the causes of this. And one thing I mentioned when I introduced the polyphenols, the chemicals that deter herbivores in these algae, is that they also exhibit strong plasticity due to abiotic factors. So we're starting to see with my current research team that things like rainfall, UV radiation, possibly temperature and salinity, basically climatic factors are very important in the phenolic concentrations. So my team of undergrads over the past couple of years and currently are starting to look at this context dependent, essentially, whether the physical and chemical defenses of Silvetia compressa varies across seasons. And we're thinking mostly about the dry season, like dry, hot season in California versus a wet, cooler season. So I'd like to show you a little bit of that work because we basically started to see that these responses in toughness, at least, depended on the sampling date. My undergrad researcher from a few years ago, Jimmy Rowe, who really got the protocols going for the Prussian blue assay, looking at the phenolic concentrations, found that it was highly variable based on the year collected. We standardized our spec curve and ran the analysis on samples that we had collected from 2020, 2021, and 2022, and found the highest concentrations of phenol in 2021. And just by like when our programs are funded and when we've been able to get out in the field, of course, these varied in when they were collected. So 2020 and 2022 were in the winter, and we tend to have, I mean, maybe they're not very wet to you out here, but we have torrential rain in LA. We get a lot of it all at once, right? And so these sampling were after rain events, as opposed to May when it's been very dry for several months. And so my student who's completing her senior capstone project this upcoming year has already collected some data on how the dry versus wet season may be impacting not only biomass and growth, but toughness and phenol creation. So I have plots of all of this, but not time to show it to you. But she's finding that there is lower growth and greater herbivory in the dry season. So even though there's more sunlight, the algae are growing more slowly. Maybe they're stressed because of temperature or something like that. The nutrient enrichment now has a significant positive effect on the biomass of the algae in the dry season. So they're more nutrient limited in the dry season, which makes some sense because you're not getting these rain events that bring lots of nitrates and phosphates. And then she's seeing this trend towards a three-way interaction of nutrients, herbivores, and the sampling date matter. So there's a lot of context specificity, and we're still working to tease this apart. And she's looked at toughness, and I just want to point you, there's a lot of variability. I think we only had 3 samples for these, but this again is puncture test. So the weight needed to penetrate that algal phalli. And the only one that is significantly positive is one that had crab herbivores. So this one had crab herbivores and was very nutrient enriched. So here we're seeing that herbivory increased toughness in the spring dry season, but in 2022, 2023, the herbivores decreased toughness. With Turbinaria ornata, herbivores also increased toughness. So I'm thinking that this might be due to the silicon in the algal phalli. So we're still looking into all of this with my current students and my summer student that I just had doing her summer internship. It is looking at the impacts of temperature and UV radiation. So to wrap things up here, there is this body of in the Fucales macroalgal species, finding these complex interactions between nutrients and herbivory that may affect their defense mechanisms. And I've typically been thinking about the defense mechanisms against herbivores. You have herbivores in the system and the algae has some defenses to reduce that herbivory. But in temperate environments, especially in Southern California, where we have really hot, dry summers, there's this possible influence of the radiation and temperature and lack of rain in these dry seasons. Whereas in Turbinaria, in the tropical ecosystems, again and again, we're finding the same interaction. It's herbivores increase the toughness, nutrients increase the toughness, and it interacts in that way that herbivores can't tougher nutrients. But you have less seasonality in the state devoted there. So my question that may keep my lab busy for many years, do herbivores ever induce phenolic defense in Silvetia, and what's going on with the silica? So I just want to acknowledge my funding sources and my students, and basically I take the little guy everywhere. There he is in the field. So with that, thank you. Questions? From what it says.
So we have plenty of time for, okay, Ethan.
Yay. I wanted to leave plenty of time for questions. I could have showed you everything my students have done, but
Thank you so much for the speech and the shout out to the overview log for the algae. And I have a broad question. So in the speech, you talk about how nutrient availability is really consequential to the physiology of the algae and therefore the herbivores and therefore the entire ecosystem. So I was wondering, like, what is the implications of that findings and consider that we're living in a time where anthropogenic changes are happening and the environments are getting more and more eutrophic. And also conversely, like as we harvest like herbivores like the tanks and the parrotfish, like what implication does that hold for the ecosystem then from the scope of these habitat forming algae?
yeah. Thanks for the question. So I think that's a lot clearer to me in the tropical example, because you have a situation where if you have nutrient enrichment and overfishing, Turbinaria is just going to take over, which it has been pretty much in French Polynesia. But in the deeper areas of the reef in Moorea, French Polynesia, they do see recovery over and over again. And the macroalgae isn't able to grow as deep as some of these large herbivorous fish are present. So I think that at least in Moorea, it's the fringing reef, it's covered with this algae. There's virtually no coral. It's all algae now. But the fore reef, the deeper areas of the reef, have been able to recover from these disturbance events and the fact that there are less herbivores that would be willing to eat Turbinaria at all. Because it, even without nutrient enrichment, it's still spiky and has lots of secondary metabolite chemicals that make fish not want to eat it. So it has essentially taken over in the shallow areas of the reef, but we have hope like for the resilience of the core reef, at least. So, in temperate environments, I think it's more murky because you know, we have such an abundance. Like the algae aren't usually nutrient limited, at least the browns, because they can just grow over time and grow slowly with the nutrients that they have. But some impacts that are affecting the brown algae, besides nutrient enrichment, are things like human trampling and warming events. And so that's why I want to go in that direction of looking at those abiotic factors and how that influences Silvetia.
Hi, I'm the other intertidal intern. First of all, great talk. Thank you. I was wondering specifically about the herbivory and how it affects the algae, and I really don't know anything about algae, so if this doesn't make any sense, let me know. But is when a predator is feeding on the algae, is it an immediate response that the algae has to get stronger, or does it have to grow like new parts that are stronger?
That's a good question. And I don't know for sure how long it takes. So the amount of time that we're looking at here is 6 to 10 days to see a response. So I don't know like how exactly how quickly the alga is responding. What I do know is that we see the response within 6 days.
We have a question from our online audience. Chelsea Gotten asks, it's really cool to see the preliminary results about the seasonality and rainfall events. Do you have any sense of what the herbivores are doing during the dry season?
Yeah. So the early work that Kira has done showed that the herbivores are able to, at least in these controlled tank experiments, they're eating more. So I think that maybe those herbivores, because we collect them from the field in the dry season, they'll be like hungrier when we get them to the lab. So I think because it's dry, they maybe have less access to like get around the tidal pools and things like that. So they might not be accessing as much macroalgae. Like they're not starving, but they're getting less resources in the natural environment at that time. So when we bring them into our tanks with some Silvetia, they're all for it, eating it up. So I'd like to look more in like with a field experiment to see what the hermit crabs are doing.
I'm convinced the questioners are doing this on purpose to make me run across the
Could you talk a little bit more about the herbivore in particular? Are they like a specialist on Silvetia, or are they just, how do they eat it?
Yeah, so I realized I didn't introduce you to my hermit crab because they're just so abundant in California that I didn't think about these things. So we were using the blue-banded hermit crab, and they're a generalist. They're an omnivore. They'll eat detritus. They'll eat other algae. They'll probably eat some of their buddies. So yeah, they're not specialists, and I don't think there are, as far as I know, there aren't any Silvetia specialists, but I would be really interested to, because we do have various snails and amphipods and isopods that are known to consume some Silvetia. So I'd be interested in looking at how different invertebrates it works, because hermit crab, they are using their mandibles and claws to like pick it apart a little bit. But if we looked at other they're not specialists on Silvetia, but they're more algal specialists. That might be interesting. These were just like the most abundant thing that we knew ate algae at our site.
Sarah, you didn't mention, I don't think, how long you were doing the growth, the exposure to the different nutrient levels. And the other piece that I had a question about is whether the toughness is, whether you tested that across the entirety of the algae rather than just in certain portions.
Right. So that stage of the experiment, the nutrient enrichment only part, was also 10 days. That's just kind of the amount of time. Pretty slow growing species. We see a response. As opposed to something like Ulva that it could grow like double in size in 48 hours in some cases. So 10 days. And we standardized to like the youngest material at like they have, you know, dichotomous branching. So we took the tip every time of the alga. So that's also something that we could look at as a different tissues and if and where they're becoming the toughest.
Basic algae question for you. So then you mentioned that like it doesn't necessarily, there's no vascular tissue, so it's not sharing nutrients or resources. So then when you're doing the nutrient addition, is that just kind of a net positive to the entire individual versus the herbivory? Is that a little bit more of like a localized effect on a piece of it? Or is there nutrient sharing that can happen within an individual alga?
The nutrients are definitely more dispersed because it's just in the water column, and any cell in the alga can presumably uptake. With the herbivores, I mean, they have access. They just have free roam of the algae, but it is more algae obviously than like nutrient uptake would be. So I haven't really thought about, well, we leave them in there for 10 days and we haven't done any like observational or behavioral type of stuff to see like what parts of the alga they're eating. My intuition is that they're eating the youngest parts, but I don't know. I haven't.
Any more questions?
I know you have a lot of questions and really cool research to fill your time for probably many years. But do you think you'll ever add Ascophyllum to as a research species?
So yeah, after seeing it, and I was like, oh, asthmaticis, that's so cool. Yeah. Yeah. If you know anyone on the East Coast that wants to collaborate, as far as I know, there's some work like this done in Sweden and Finland with Ascophyllum, but I think most of the work I've seen was more like population genetics. I'm not totally familiar with the Ascophyllum research, but I'm pretty sure that these type of defenses happen. Yeah. So
all right, I'd like to thank Sarah Joy for again giving us such a great introduction to the macroalgal systems. And yes, it sounds like you've got the rest of your career set up with a variety of different questions.
Back to the Trump.
Yeah. So thank you so much.
Thank you.
Next week's Rock Talk will be our very own Kyla Bennis, who will be presenting what's happening on the rocky shores around the Shoals Marine Lab, stories from over 40 years of SML intertidal surveys. So please tune in next week at 8 p.m. And as a reminder for anybody who's here now and will not be here in the future, you can always tune in. Our programs are live streamed, and you can also join us by seeing anything on our YouTube channel. Thanks for a great Rock Talk. And thanks for everybody being here.